Come to the presentation of plant genetic resources in India, management and utilization in genomic era. Once I again welcome, and first of all, I request our Honorable Vice Chancellor to welcome by presentation of bouquet of flowers to Dr. Kuldeep Singh, Director NBPGR. Now I request Dr. A.K. Gupta sir, Dean and Faculty Chairman, to present a flower bouquet to Dr. Ashok Kumar ji, Head Young Class Evolution. Now I request Dr. Sunita Gupta ji, Director PME, to present a bouquet to Madam Rasmi Yadav ji, Principal Scientist and BDGR. वैसे कुलदीप सिंह जी किसी परिचय के महताज नहीं है, लेकिन इस समय जो लोग हमारे ऑनलाइन जुड़े हैं उनको थोड़ा सा मैं इनका ब्रीफ इंट्रोडक्शन करूंगा, क्योंकि वो यहाँ के अलुमनाई है और आज में बड़ी खुशी है कि वो आज अपने अल्मा मेटर पे पहुंचे। इससे बड़ी खुशी और कोई नहीं हो सकती। डॉक्टर कुलदीप सिंह जी born in Kashmir, J&K state on 25th December 1960. He did his BSc from Supadia University. Then he did his MSc and PhD in plant breeding from BU. And he was decorated with Sardar Iqbal Singh Dhillon Gold Medal for securing the highest mark in his post-graduation program. And he was started his career as an assistant wheat breeder and worked on wheat since July 1990 to 99, and during this period he was associated with wheat breeding, release of varieties, and he has released 3 major varieties of wheat, that is wheat BW 443, 343 and 396, and these varieties are highly suitable for water stress conditions. During this time he has also established a protocol for haploid development in wheat through wheat and maize process in wheat hybrids. Between this, he has in 1992-95 he has joined International Rice Research Institute, Philippines, for his postdoctoral fellowship under a World Food Laureate, Dr. G.S. Push, and completed his postgraduate fellowship, where he has developed a complete series of secondary trisomics in rice. And from 1992 to 2015, he has worked in different capacities as molecular genetics, senior molecular genetics, and director of School of Agriculture Biotechnology. During this period, he has received a tune of more than 50 crore rupees by externally 30 externally funded projects. And during this time, he has developed 4 varieties of indica rice through marker-assisted selections, and 3 varieties in case of basmati rice. And out of them, one was the country's first semi-dwarf variety and resistance against bacterial blight in rice in basmati case. That is a good achievement of Dr. Singh. And during this period, he also identified 2 genes for stripe, 2 genes for rust resistance, 1 gene for powdery mildew, 1 gene for cereal cyst nematodes from diploid wheat and transfer into the hexaploid wheat. That is also a great achievement of Dr. Singh. Later on, he has credited more than 130 papers in prestigious referred journals like Euphytica, Day, Science, Plant Breeding. He has also guided 15 MSc students and 14 PhD students. But out of them, namely Mr. Dharmender Kaur, Amandeep Kaur Sindhu, Pallavi Malik and 2 more got the highest international scholarship, that is Monsanto Behag World of International Scholarship. And this indicates the depth of knowledge and kind of research. There is no plant breeder who has such kind of number of the students which have got the such kind of the international scholarship during his tenure. And after 2016, Sir is holding post of director NPPGR, and till date he is continuing. And he has special privilege to this institute. After joining our Honorable Vice Chancellor, in last October, we have submitted a project in collaboration with NPPGR for transfer of pest and disease resistant from wild relatives through use of genomics. And this is the only project that was selected from agriculture side to present before the project appraisal committee. And in April, Dr. Singh himself presented the paper in front of the PSC, but however, due to the certain conditions of COVID, the matters could not materialize. Later on, we have also prepared a project on skinning of chickpea, especially for dry root rot, as well as water stress and winter hardiness and heat terminal. And recently, we have submitted under the leadership of NPPGR a project on management of orobanki through intervention of genomics in National Agriculture Science Fund. Sir has also provided 250 system genotypes, and for that they have today morning visited the post-him. And in before 4 days, that is on 1st October, we have also made a material transfer agreement to receive 50 genotypes of chili to screen against virus. And we hope that we are asking only 50, but sir will provide more than, so that we can evaluate them properly and can get a good material for resistant against viruses. And I hope that under the dynamic leadership of our Honorable Vice Chancellor, this process shall continue, especially in those crops which are highly valuable for our state, especially malt bean, mung bean, sesame, and underutilized crops. Thank you, sir. Once again, thank you on the holy place of alma mater of Dr. Kuldeep Singh. Thank you, sir. Now I request our Honorable Vice Chancellor for welcome of the dignitaries of NPPGR. Welcome, sir.
Record for introducing Kuldeep Singh, we welcome you. Dr. Ashok Kumar, Madam Rashmi, and my dear colleague, and all those are online listening to today's program. I think we are quite fortunate. A few days back, Dr. Roda has recommended to invite Dr. Kuldeep Singh. Why not to have a lecture of him? Right? We have a recommendation of such a tall authority. Definitely, you are such a person is recommending that this. Need not to introduce much about that. Definitely, Dr. Kuldeep Singh, our whole team is welcome. No doubt, जब अपने घर में आते हैं तो दीवारें भी अच्छी लगती हैं, तो वो अच्छी बात है. और if you coming purposefully, I think that make a good sense of that. And because we are colleague at PAU, and we know many things working together, kind of working atmosphere there and working together, going across what I usually say, have a linkage with more than across the subject. No doubt, in PAU, genetics was pushed aside, but anyhow, they made their place within the university. And definitely after, we know that when I joined PAU in '85, we have just a small laboratory of tissue culture there. And that is very much near to pulses section. And from there, they have grown such a full-fledged of institute of a directorate of is a school of biotechnology within the university. I think that kind of efforts they make at the institution meeting has come up, and because while I was at ICR, so I was a frequent visitor to PAU, particularly the kind of the biodiversity program he took at PAU in both wheat and rice. I think that kind of material nobody having across the world, I don't think. I can say we can endorse that way. The kind of material I saw in the field. And he making all kind of things how to make use of the young. And I don't know if he is a biotechnology, how he can fit into NBPGR. I think that working on that as well will bring to back in such a, which is the NBPGR have largest gene bank in the world. And heading that institution itself is a pride of everybody who are in it. So I'm not saying much about that. When I was there, the collection was 3.9 lakh something like. And now it is across 4 lakh, maybe 420 or 20 like that. 4.6. I think that keep on adding on that. But beyond that, I think what the gene bank is required, I think that task you might have completed are still there because that system was very old, and we are trying to hold that one. That is big task. And besides that, he has given a new dimension. New dimension how the genomic resources. Genomic resources is not much known how to document and how to make use of that and bringing that NBPGR in that direction. Because we, as a conventional breeder, we target the whole plant and the recombinant. But when talking of really in today's science, so where is actually the gene is located and which gene is important to designating that is becoming now more and more important. And the trait-specific gene identification, I think, is the biggest task now. And he is making all efforts. No doubt, Laksa might be knowing more. We are the hotspot, one of the hotspots. And we remain the underutilized. I also admit that. Because other day, Dr. Kuldeep Singh giving a lecture from the Washington State University. He said that he has developed a genotype which can survive in the wheat, particularly in 45 degrees. Why not to have people collaborated with Jordan? We can provide the similar conditions. We can provide even the sub-zero condition and the highest condition. So in that temperature variation and these things all, and then other all extreme temperature, extreme disease condition, all these things is the key for making the improvement now in the science. It's very difficult from thousands of lines, which line is important. Unless you not identify that one, it's all listed of resources. So hotspot they have their own importance. So not emphasizing on that much, but Kuldeep Singh is well versed with the system internationally, nationally, and so many lectures, many places. And what I can say, the first marker system selection variety, PR 106, is the first work as recognized in the PLU that did his with the marker system selection. So that's not taking much time because NBPGR, I can give you a little bit more because that institution is almost like our university. That has 11 stations or 10 substations across India. And everywhere they have a presence. So like that is heading such a use, and scientist strength is 153 or something like that. So I tried to reduce that one, but not able to do that. because there is a strength critical. I think 147 was in place already, so there is not much scope of that. But I was going the strength of Okay. That's how you are most welcome. Welcome to this place, and it is known to you. I am really happy because that Pradha is given a lectures very much on bioresources, and I think that was the kind of first title like that. The bioresource leads food security and nutrition security at this time. So, I think in that series you will add further value in that. You are invited to here to and address our colleague, and we want that not we going deep into the science, but how to reach to that science. I think I request to you that. Thank you, Dr.
Honorable Vice Chancellor, Dr. Singh Sandhu, Director of Research, Dr. Zakasa, Dean of Agriculture, Dr. Haji. First of all, thank you very much, sir, for giving me this opportunity to be here back to my alma mater after quite a long time. Left this place in March 1983, and we had an opportunity somewhere around 2012 or so, where I could contact some of my friends in Jaipur, and we came here, but that was the month of June. College was closed. We could go to all the buildings, touch them, but there's no one here. Really thankful to you for giving me this opportunity to be here with you today. When we were students here, never realized that what the importance of this, how this was developed, how this all happened. But when you grow, you realize many things exist. I would like to convey through your office our gratitude to Thakur sir, who was such a visionary. At such a time, he established a college in a very small hamlet. It was not in a village. How was his vision that really keeps us thinking that how visionary this person was? And please, sir, from our side, through your office, we will like to convey the gratitude to the family. It is because of them that we are here today. We had that opportunity once to go to their palace as well as guests. How humble he was, and then two of his sons were also studying here. Sangramji was here, and one. And I think I'm told that both of them are also superannuated from here. But this is a very, very important thing. And one more thing that I would like to add here, that when I was nominated by ICR for University of Udaipur, went to Udaipur with the Vice Chancellor, and presented my documents. He asked me, "मौज मस्ती करनी है या पढ़ना है?" Okay, जी पढ़ने के मूड में आए हैं. कहते हैं अगर मौज मस्ती करनी है तो यहाँ उदयपुर में रह जाओ, पढ़ना है तो जॉब में चले जाओ. I didn't know what the job there is. So were good people at the time. A vice chancellor sent someone to the radio station. They booked a ticket for me, and I was packed back. The Chetak Express. In early morning, I was here, and I reached the same office. Dr. Mehta was there. She was Punjabi speaking, so थोड़ी सी मुझे एक शांति सी मिली थी. Okay, this is not an alien land here. And so I think with that one we started here, and every teacher was so good, so kind to us. Very, very good, very important. The teaching, I think, sir, I won't hesitate saying this college had the best teaching in the country. That was sort of, I was just telling Dr. Jain Sabde, always smiling, the teaching was so good, and that was animal sciences, and we were taught so widely. I know endocrinology probably more than anyone medical science student knows. That was the level of teaching here. And I could maintain 4 out of 4 here. So me and Abhay, we were 2 students who could maintain. I think we were after a gap of 3 batches, the 4ers were. We never knew it what the 4er is, but when first semester it was 4er. When we came back from the holidays, so it was something that, okay, we had done something good. Then I think it was Harish ji. Dr. Harish was head horticulture. It will be एक semester तो बहुत लोग बना लेते हैं, बात तो ये है कि इसको maintain करोगे. That was a challenge he gave to us, and there were many teachers. जो Dr. Bartla was there in dairy sciences, जिनके पास 4 महीने maintain करना बड़ा tough हो करता था, but I think that was something. This institute gave me beautiful things in my life, and that was the base we. Rajasthan has made tremendous progress. And sir, probably when you were appointed the Vice Chancellor of this university, सबसे ज़्यादा खुशी मुझे मिली. इससे ज़्यादा किसी को नहीं. और से कह सकता हूँ कि I knew that college was going down because in between there have been changes, things keep on moving. But when you took over, I knew you were working because I started working with you right when we were at PAU, and then I worked under your supervision at NBPGR when you were deputy director in the crop sciences, and I will show you the way you supported that has brought fruits as well. The new gene bank is completed. We'll show you that one as well. I know how good friend you are and how hard taskmaster you are of all the things. And this for us, in fact, to realize. And I'm sure under your leadership, John University will go much higher. So I can go back to my talk, how much time I will have, so I can then do it accordingly. 1 hour? Oh, good. So how is the control, the presentation? Yes, yes. You can, maybe you can do it on the presentation board. Good. So the title I chose is, and I will have control of shifting it or some. If you do that control, I'll be very happy. I can go there. Well, so I've not put too much of science in it, but maybe at the end of the part, if the time permits, I can bring in really hardcore science part into the topic as well. But this is probably more, I understand we have the undergraduate students, we have the postgraduate students, we have the faculty. So the presentation is according, but then I will like to do some. So this is biodiversity definition. It encompasses 3 things: the ecosystem, the diversity at the species level, and diversity at the genetic level. And what I will be talking with you today is the last part. That's the diversity and the genetic diversity. I will not talk about the ecosystem diversity, about the biodiversity, and the global jungle program I talked last time. This one. The biggest challenge, in fact, to the biodiversity is our population. And we all might have realized that the COVID, which was a pandemic, but it has yielded some good points as well, that we realized that we were actually misusing or over-exploiting our resources so far. And in fact, the major We can see right now when we were the students from Jaipur to Jogni, there was hardly any village, anything in between. And today I didn't see even a 10-meter span which is free from the construction. So I think that is really, humans are a major source of biodiversity. There are huge challenges. These challenges are in terms of the biodiversity. First is the climate change. In fact, out of the 23 hottest years, 21 were in last 23 years. That means from 97 to 2019, for which we have the data, this is the one. See the extinction rate of that has gone 1,000 times. And very important data that the population of the vertebrates in the last 40 years, from 1970 onwards, it has gone up by 60%. And last one, 75% of the Earth's land is substantially degraded. These are the challenges, and if all this happened because of the overexploitation and the human populations. Good. Fine. And in fact, the genetic diversity is really very, very important. And if we are aware of the Sustainable Developmental Goals of the UN, to which we are signatory, there are 11 SDGs with which are 12 of them. Agriculture is directly involved or responsible. But out of those 6 are the ones where plant genetic resources or plant breeding is directly involved. Be it no poverty, be it zero hunger, be it good health. For everything, this cannot be achieved without plant genetic resources. That's the important thing we really need to do. And to share with the students here, it's not something which is in the air. We are providing the quantitative data every year. And NBPGR has a responsibility to support the data from the country. We collect the data and we support to the nation, to the global world, passing all this data. And we are actually participating. So this is all quantifiable. Okay. And my topic, which is plant genetic resources, it basically involves 4 things. That's the collection of the germplasm, the conservation of the germplasm, the characterization of the germplasm, and of course, the utilization. All the 4 of them. And another fact with us today is the population. We are right now something around 1.33 billion. We expect it to be 1.44 billion in 2 years from now or 3 years from now. So that's the time when we equalize China. By 2030, we'll surpass China. And by 2050, we are expected to really, maybe we will stabilize at that point. So that means the question for us is, will Mother India be able to sustain 1.66 billion people? That's our major question. And we are convinced that to feed this one, the genetic resources will be the genetic resources will be what breeding approach you follow, what you do, but we believe that the plant genetic resources will be key to achieve those ones. See, what are the difficulties we are facing at the moment? How the things have changed. This is one of the major paradoxes in agriculture today. Let's say maybe 100 years back, we were dependent on around more than 37,000 species, which the humans were using at one at a time. But what's happened today? 60% of our calories were derived from 3 crops, wheat, maize, and rice. So that has actually led to decline in the general biodiversity. And I will show its impact, how it has impacted the global population of the country. So that was a general goal. See, just within India, after the Green Revolution happened, I don't know whether you'll be able to see this one, there has been a general decline in the use of the millets and some other important species. Almost some of them are 40% decline to 30%. And there has been increase in some, of course, soybean data may be slightly biased because we introduced soybeans somewhere in the early 70s in the country. So there's a huge increase. That may not be a real data in the perspective, but we see the lower one. See the palm oil, see the coconut oil decline, and see the increase in the palm oil. And coconut oil is no way inferior to the palm oil. So there have been some policy change that we are actually doing. And let's take this one. Now see here how the things are changing. See the figure on the right hand side. This figure is, which is a family health data. It's a national data which was collected from 2006 to 2016 and published in 2018. I'm taking the example of states like Punjab. See Punjab. Now, Punjab is not a poor state. We have the most progressive farmers, highest productivity in the world, almost 12 tons per hectare is the average yield of the grains, and another 12 tons of the straw. That means 24 tons you are producing every year. Then why? Why there is an increase in the proportion of the population which is anemic? It increased by 15% in that one decade. And the basic reason, in my opinion, is the monoculture. Because 20 years back, there was still diversity. People were growing different crops. Now it's wheat and rice. Almost 70% area in Punjab or Sindh knows it as a wheat and rice. And I simply say to farmers now that 20 years back, a farmer who was going to his farm would come back eating something from the farm, maybe some minor fruit, some vegetable, or if nothing, sarson ki gandal jo hai, usko kaat ke chook dete the. That is a rich source of iron. We have gone to the monoculture so much that a farmer, if he goes to the farm, can neither eat rice nor he can eat wheat. So whatever he's getting, selling them into the market, for getting money, of course, but he cannot pay back to buy other things from the market because they're so expensive. And those are the reasons. There has been some better thing in some of the states. So in fact, to tell you that we are seeing the diversity. Now this diversity at the crop level, the reduction has been very significant. Earlier, we were depending on a very large number of the crops, but now, unfortunately, we are actually going. And that is a concern for the nation. Have a look. I mean, last year, 3-4 global papers came with very good analysis. If you see the right-hand lower side circle, the circle gives you that what is an optimum food one has to eat. You can see the left top circle, that's I think the North America. They're exceeding most of the things, with the cereals, with the meat, everything. But see in India, we are eating cereals a little more than we actually need it. But for all other things, we are eating too less. Be it vegetables, be it fruits, be it nuts, whatever it is, that means our diet is not at all a uniform or a balanced diet. And that will lead to a lot many problems. And this is actually the role what agricultural scientists have to think how we can ensure, because we see now a large population is wasted, I mean low waste, undernourished. 40% of our children below the age of 5 are protein malnourished. Almost 60 to 70% of the rural women are anemic because of the iron deficiencies. So those things cannot be actually covered only through wheat and rice. If we see globally, probably I was also working on biofortification of wheat, but if I have to see right now, probably that's not the right way. We need to really popularize those crops which are already richest in source. You see, Karonda. Karonda is one of the richest sources of iron, and it's a bush growing along with this one. Can we do something, sir, onto this one to popularize Karonda and really make Karonda available to every household so that our women are not anemic, our men are not anemic? We got some good germplasm as well. Faizabad se we collect together. Jaise aapne pichli baar kaha tha, sir. We send 1% to Faizabad. So this is how you actually, the visionaries. I was there, and Sansa told me that Karonda is very good there in that region. So when next year season we organized an exploration, and one of the Karonda is, color is like apple, and size is something like this. We have already collected it, it's planted there, and maybe we will evaluate it. And so these are the things probably we agriculture scientists have to think in terms of the national, because there was a time when food security was important, cereals were important, giving them or feeding the bellies was important. I think the time has really gone now. We have to see where we are globally and what we need to do on this one. And this is again the left side. I don't know, this may not be visible here. Yeah, but what to tell you simply, the left side is what's the energy requirement of different crops today, and what's expected to happen in 2050. So right now, around 50% of the calories globally we are drawing from our seeds. There may be a little bit difference in the data. Some say 50, some say 60. But what's more important that even in 2050, around 40% of the calories which we will still derive from the cereals. So that means we really need to see what sort of food we should produce, what sort of the varieties we should produce in different crops, how we should really diversify our systems to be sure that our population gets a balanced food, balanced nutrition. And we might have seen there's a huge surge in the medical sciences, good number of hospitals coming up, and at the same time, our expenses for medical are also increasing. And increasing sometimes to the level that most of the families cannot afford. And a lot of that has gone because of that imbalanced nutrition. So that's probably we need to play a role. And in fact, it's based on this one. NBPGR's thinking is sustainable agriculture and diverse food intake. Both are equally important for our health. So we are actually now working on this one, and the figures, the photographs I have given here, you may be familiar with some of them, may not be familiar with some of them. But they are in different areas. But they are all very nutritious, and I will tell you that we are working on some of them. So that was a sort of a negative point, but at the same time, we are blessed, India is blessed by the nature that we are one of the very richest regions when it comes to diversity. There's diversity be it in plants, be it animals, be it microbes, be it fish. We are very diverse nation. So Mother Nature has bestowed a lot on us. It's for us to see how we can really use it. See, for example, almost 1,000 edible plant species are there. And how many of them are right now in use, as we have seen. We know out of the 8 Vavillo centers, 1 center falls in India. And this was realized, and globally, right now, 4.8 million accession of around 1,600 genera are being conserved in around, I think, a good number of 700 more odd gene banks in the world. So this is, and have a look here at the gene bank, NBPGR. So we are one among the 20 gene banks who have more than 50,000 genetic resources. though we are number 2 in the world at the moment. And genetic resources, what we have done, classical examples, which are about the green revolution, 2 genes, which all of us are aware, one dwarfing gene in wheat and one dwarfing gene in maize about the revolution. But there are other genes from germplasm from India which have actually, I won't say revolutionized, but has actually sustained world agriculture. See, the first example that comes for rice. Grass-stem virus was such a devastating disease in the eastern part of the world that I think in the early 70s it was thought probably rice may get eliminated in that region. Until Dr. Kush identified a resistance source in a wild species Oryza nivara, and this gene gave a sustainability to the rice production. And this gene is still effective. Only gene known so far. Others, some of the examples with the US, for example, the downy mildew resistance in muskmelons, the green bug resistance in sorghum. If you see the wheat of Canada, Canada is one of the major exporters of wheat. We may be importing something from them as well if you follow. When for this Irish famine, when they immigrated to Canada, most of the wheats they took from Europe, they were highly susceptible and very poor quality, until someone got a wheat line, what's called as hard red Calcutta. This line made a basic genetic stock for disease resistance and quality of the anything. Very well documented by them as well and acknowledged. And a recent example of submergence tolerance in case of wheat, I will show you some photographs later on to this one. This gene has given probably more returns to the farmers than the BT gene in cotton has given. Now, if we see the economics of that one, Monsanto might have got billions of dollars from India for that one gene. India did not ask him for a single penny from anywhere in the world. Now, this is, I'm sharing with you, but this is very important for us when we are making the presentation of the international platforms. There are a lot of criticism that India is not sharing with genetic resources, but we need to quote some of the examples here to tell them what India has given to the world. These are some of the examples, and all these examples are documented in textbooks now. They're not only in the research papers, they're textbook examples. And the younger colleagues who are listening to this lecture, to tell them that until 1992, the genetic resources, plant genetic resources, were the property or heritage of the humankind. So that means you can go to anywhere in the world, get it, use it, and anybody can come to your country, get the genetic resources, use it. But after 1992, when the CBD came into being, into effect, thereafter it is the sovereign right of the individual country from where the genetic resources are. That means genetic resources existing in India, nobody can really use or misuse them without the consent. So there are some good and bad impacts of that one, but to tell the younger colleagues that we are actually signatories to it. There are these 3 important international instrumentations, treaties, what we call CBD, Cartagena Protocol, and International Treaty on Plant Genetic Resources of Food and Agriculture. India played a very, very important role in these ones. India is signatory to these ones. And to safeguard these international instrumentations, we have the 3 national legislations and national acts called as Geographical Indication and Goods Act, the Biological Diversity Act, and Protection of Plant Varieties and Farmers' Rights. These are our national legislations to protect the international instrumentations and ensure that our genetic resources are not misused. They are exploited in the right direction. Our farmers are protected. The genetic resources they identify, they are protected. These are the important instrumentations. And why I'm presenting, because NBPGR is a major institute supporting all of these. Of course, that has the responsibility. For example, ITPGFRA its responsibility lies with the Ministry of Agriculture and Farmers Welfare. And all this scientific backstopping goes from NBPGR for this one. For others, CBD, Nagoya Protocol, is the Ministry of Environment and Climate Change. It's their responsibility, but scientific backstopping, a lot of that from agriculture sciences goes from NBPGR. And to tell my younger colleagues again that probably India realized it long back the importance of genetic resources, and ICR is the only organization in the world which has such a beautiful mechanism, having 5 bureaus in the country, each responsible for plants, animals, fish, micro, and insects. Nowhere in the world you will find such a good mechanism. And we have a responsibility of the plants, so I'm presenting one of the organizations, that is National Bureau of Plant Genetic Resources, but 4 others are equally important, and they're at different places in the country. And what are the plant genetic resources for us? It may be the land races, it may be the wild relatives of the crops, it may be the release varieties, the genetic stocks, the farmers varieties, the transgenics, everything, whatever it is, those are part of the plant genetic resources. And this is how NBPGR has actually evolved. Although genetic resources in India, working with them is as old as IARI is. So we have been working with them from 1905 onwards at different capacities. But until 1976, we were a partner. There was a division at IARI, and that division was then elevated to an institution level, but later came to be known as National Bureau of Plant Genetic Resources. And Dr. Paroda played a very, very important role in developing the infrastructure. Not here, all the 5 bureaus, in fact, it's all his vision, his thinking, and all beautiful infrastructure. So we had different phases. As the world grew, as we also continued to grow. And if you see our nowadays, in fact, the impact, we are looking, because we have already partnerized our gene bank. Thank you to Dr. Sandhu sir, who has been very, very supportive, because getting the money from ICR is not that easy, but if you have a good score from scientists like him, you get it, and we got it. We'll show you how we have to do facilities. We are actually now using the modern sciences of genomics as well. We are actually thinking of putting these land races and crop varieties in large scale use, and I will show you how we do it. I will now take you for a visit to the NBPGR's activity. So plant exploration is our major activity. Every year we conduct around 35 to 40 explorations in the country. And I invite you, if you feel, although we are very confident that Rajasthan has been covered fairly well when it comes to collection of genetic resources, because Dr. Paroda was heading it sometimes and the folks was there. So, and the desert was most important. We have a regional station at Jodhpur as well. But still, if you feel that some of the sources, some of the species which have not been done, which need to be really more collected, please let us know. We will collaborate with you. We will take and get this university on board and do more collections on this one. And see, as of today, we have something 4.6 lakh genetic resources in our genetic resources in gene bank, out of which around 60% is the indigenous. So that's something now. This 2.75 lakhs is an old figure. We have crossed 2.9 lakhs now. And our emphasis nowadays is more on very difficult areas like Andaman Nicobar, Arunachal Pradesh, Leh Ladakh, all these difficult areas which were not covered earlier. And we are getting really all sorts of genetic resources from them as well. This is how our people go to different places, talk to people, not only genetic resources, the traditional knowledge associated with these genetic resources is also recorded and is a part of us. This is for the students who are youngsters, beginners, that we really Whenever we collect something, we have a passport data. So nothing will go to gene bank without a passport data. That's the minimum which is required. If you go for collection, we have NBPGR has those basic norms that someone has to collect it, what you should note about it. Anybody can undertake the exploration at their own as well, but please go to this site and see what are NBPGR's minimum descriptors for the passport size data. So that if you have this data, this can go easily. I will skip here. Okay. Exchange of the genetic resources is very, very important for us. So here we really have to take care of both biosecurity and our needs as well. And on an average, we are facilitating the exchange of something to 175,000. Each sample individually being purchased. And this is what plant sciences are doing. This is what we missed in humans, and we have a corona pandemic. And if such a laxness was there in plants, we would have several epidemics in the crop plants. But fortunately, the system is so robust. You have an individual division there who do analysis of each and every sample. We are aware that nowadays things are changing a lot. People are not getting phytosanitary certificates, so we really have to support our colleagues in the country. But what we are doing, and this is what I'm asking many times when you introduce germplasm from outside the country, if the provider is not giving you the phytosanitary certificate, we have to grow that for one season under post-entry quarantine facilities. So that may delay your seed arrival, and we cannot risk onto this one. This is one area where Director NBPGR is not under the control of the Director General ICAR. He is under the Quarantine Act. So nobody can influence him to really allow for any laxity in this case. So very, very stringent in this one. But we are developing a lot more facilities, collaborating with all the institutions, ensuring that everyone who wants any germplasm from outside the country, they get it, and we get it really clean, and we don't bring in any unwanted disease or any unwanted pest into the country. Once we have this one, we are conserving the genetic resources in different ways, what we call as medium-term storage, which you conserve at 48 degrees, which is our working sample. Long-term conservation, -18 to -20 degrees Celsius, cryoconservation, in vitro conservation, and the field conservation as well. And in fact, to tell you, this is March 31st data. 3,45,920 germplasms. So this is what you supported NBPGR and completely, it's all a good management system, very well connected. So anything happens, the people who are responsible, they will get a message on their phones and everything. This is very simple. But this is completed before the COVID happened. We were successful. Thank you very much for all your support you provided in NBPGR. There are several species which are recalcitrant, where seed cannot be conserved, and there we are actually conserving them in into cryoconservation. There are several species which do not produce the seed. We conserve them either in the field or during in the in vitro culture, which called cryoconservation. So this is the status of our in vitro gene bank and cryo gene bank. And so we are actually stressing nowadays not on potent conservation, especially for horticulture species and wild species, which we may lose it onto that one. We are emphasizing on conservation of potent, so that can be used at any time later on as and when required. We have a field gene bank as well, and here different institutions. We have collaboration with 59 centers in the country who we call as the national, our NAGs, national biodiversity sites, sorry, national active germplasm sites, 59 of them. And if you feel any of the genetic resources are important to this region, and you can provide the support, the land, other things, and that gene bank could also be established here as well. Any resource which is very important to this one, very easily can be put in this one. And this is to tell my colleagues that we have this information in different forms. You want the pearl genetic resources, we have almost 8,000. You want something to know about the castor bean, this one. You want to know the flower bean, a mung bean, whatever it is. So, for example, mung bean, around 11,000 genetic resources are available. This is about the vegetable crops, different vegetable crops, you have the different sources available. All these sort of, even for individual land races. So, this is CSM, we have something, how much? Almost 9,000 accessions available. For safflower, almost 7,000 accessions. And safflower is also very, very important. I don't know how this region is performing, but it's a good source of oil. So, and these are the 4 species actually here: sesame, safflower, linseed, and Niger, on which we have a mission-made program right now, which is a DBT fund project. And each of them getting a good support. I think sesame got 32 crores, and safflower something also 28 crores or something. Linseed 25 crores, and Niger 100 crore funding from DBT for these 4 crops. So overall, we got around 280 crores funding for 13 crops. This is this one. And to tell my colleagues here that all these genetic resources where we had a good information, we have georeference, and they are all available here. For example, if you go to this our site, NBPGR, this is a site here, anybody click here, you want for rice, these are different here. You want to really click on any of these spots, it will give you what accession it is. And what we are doing now is actually superimposing the soil maps onto it and the climate maps onto it, so that anyone who is interested in any type of germplasm, they can look for themselves at what germplasm they are looking for. For example, for sesame, you asked us for any genetic resources, but maybe a few years down the lane when we have completed, for example, 8 or 10 species we have already completed. One can straightway go into this one. One can straightway look on what you are looking, click onto this one, have the accessions, and then the intender can really identify what they are looking for. Likewise, utilization part, because there's no need for, only election may not serve our purpose, though it's for posterity. But we are really looking at how the genetic resources, which are the gene bank, can be used by our colleagues in the country, the breeders, other researchers. We have really started the large-scale characterization. And the 2 projects which went very well earlier was wheat, where all genetic resources, all wheat was taken out, evaluated at one place, and of course the chickpea as well. But we have a slight change in this strategy. At the time, other things were not available. We'll complete okay. Go to this one. So, along with the characterization, we actually evaluate these genetic resources as well. We have a good CRP program. That's a real good one. Again, thank you very much for that program, which were conceived under your leadership, sir. And a good number of genetic resources are being characterized. And for example, this I have given some examples. Chickpea seed, our data shows that root rot, there are 21 genetic stocks which are resistant to it. So anyone who's interested into this one, really go to this one and then see, validate it, whether they are really true or not. We say, because this all data has been generated by experts in the country. There may be, for example, we know that for dry rot, it might have come from one place. It may be resistant in one place. It may not be resistant in the second place. So the individuals have to see if there are 21 of them. One is sure that one or the other may be resistant at one other place. So this sort of information is actually accumulated. And our databases are getting richer and richer for this purpose information. Take up anything with individual accession-wise data is also available. Anybody can really look, for example, white rust in case of mustard. These are the accessions which are already resistant. For F8, some of them. For alternative light from this one. For example, for okra, the bindi, you have the yellow mosaic virus resistance, or the elation virus resistance. These are accessions which are already. So the individuals who are looking for this one, look for the information which is available and procure the genetic resources accordingly. And happy to say that this has been always that use of this one. Dr. Ashok Kumar has done some analysis. We are doing more details of this one. दस अब कितने 2000 में से कितने लाइनें हैं varieties जो हैं इसमें से 900 से ज़्यादा है. Out of 2000 varieties released in last 10 years, 900 of them are direct selections from Genomics. And this is I have given from one state, that's Telangana, where they have released 6 varieties just from selection of the Genomics. And we are happy because they acknowledge that this has been done. They have to release the varieties, but for NBPGR it becomes, yes, the material was there, it has been used, the varieties have been developed, and the germplasm is used. So there was no conflict into this one. We are happy because they acknowledge it. So this is what we are actually looking from whole of it. Use it, acknowledge it. We know that because we really have to prove. When Dr. Sandhu was there, he was asking how it's being used. We have to answer it. And unless we have this type of data, we will not be able to survive ourselves. And this whole information is available in the PGR portal. Exchange of the genetic resources. I think largely people are saying that the genetic resources are not being used. But so this is the data. And this is the data where you get the indents and send. Not that material which NBPGR has voluntarily sent to different institutions for characterization. Excluding that one. If we use that one, then it has gone in the last 5 years more than 67,000 or so utilization. So this is how NBPGR is sharing the resources in this release. And just to give you some pictures to depict the variability in different crops, because these are important things to know. This is rice in Manipur, be it black rice, or be it olive green rice, be it red rice. You have all the, see the color of the blackness. And these are becoming very important now, more demand coming for this type of the genetic resources now. So beans, again, from one area, Singh. This is the wild species of cucurbits, which has been very, very important for giving many disease resistance genes. Variation in the roots. Variation in the wild species of different Solanaceae. Some of them are being used as the rootstocks for fusarium, especially, where you have the root, the soil-borne disease problems. These are really becoming very, very important. This is a fruit from Rajasthan, a minor fruits, and I'm sure, sir, Rajasthan has a very good number of the wild fruits. I think that's an area probably we can join hands and collect them, because we may otherwise lose them fast. See the variability in bark. We have a good genetic barley germplasm. I think Rajasthan is one of the important states for protection of the barley. Who's asking people, can we go back to Sattu? And why not to use the black, which has a very high anthocyanin content? You have the varieties which are less anthocyanin rich, and of course, one can see the other things as well. So we really need to see, you have an agribusiness center, some of the youngsters can really start this type of the work as well, which could be a contract farming, and go maybe pack Sattu instead of what we are going up gutka and other things. This could be a sort of thing, we gutka ko chhodkar Sattu jo hai wo bahut rich hai. And people will definitely come back to these things. Very important, for example, very unique genetic stocks which people have collected and available. So if anybody who's interested, looking for any sort of genetic resources, you will find them all here in the PGR. We are working on some of the these minor crops as well. be it buckwheat and amaranth of the hills, chinopods and jobster from eastern India, some of the legumes. This is an important thing, sir, which I would like to share with you. And people here, they know this is kalangla, which is a crop of the deserts. But 50% of our requirement we are importing from Africa. We are not even able to produce whatever is required. And we had an opportunity to visit some of them, some one of the industrialists in Gujarat 2 years back. He was kind enough to tell where they are used. Jitna kaju kari hai, kaju barfi hai, sab hai, this is the substitute. You don't get kaju, and this is actually what is being used. But the good thing is, this is more nutritious than kaju is. It's huge. And Kazri has released 2 varieties last year for this one. And that's probably we really need to look into this type of this as well. So ICAR-NBPGR has a responsibility to work on this one as well. So that was on genetic resource availability, but better to be used in global criticism is to be used in. Despite I showed that how much germplasm ICAR-NBPGR is using, but still, yes, there is, for example, IRIS gene bank, which is one of the important gene bank, very well characterized gene bank. Even less than 5% of genetic resources have been used here as well. But things have changed now. And these things have changed because of the genomics. And I will show you how. See, this is one gene, how genomics has changed the things. This is one submergence tolerance gene. This gene was known to us in 1948. And breeders could not really develop a high-yielding submergence tolerance variety. Why? It's not that the breeders didn't do it. They might have done it, back crossing, Globicare, but nobody. The reason was, when the gene was mapped, and this gene was mapped here, and I'm happy to share that this was my postdoc work in Erie, where we mapped the centromere position. The centromere in chromosome 9 is very subtulocentric. And the gene that was conferring resistance to the submergence, that actually got located. And if rice genome was not sequenced, we would not have been able to clone this gene. And when this gene was cloned, this whole region was identified. Along with this good gene, submergence tolerance gene, there was another gene that is actually impacting yield and other things. Until it raised a very large F2 population and broke that negative code with this linkage. And once they had a genetic stock that was free from that disease, that negative gene, then it became a history. And see, you have submergence tolerance gene. And this gene is contributing immensely to the global economy, especially in Northeast and other things where Eastern India, where the submergence is. So this is first example of how genomics could really get into the practical use. And I'm saying everywhere, if rice genome was not sequenced, we would not have been able to really get that. Because our understanding was not there. Skip some of them, we are not clear to. This is how we are now started a complete model. We are not going for the piecemeals. We are actually taking complete germplasm out, phenotyping each one of them at minimum 2 places, and genotyping that. And then we have an idea that we will generate, we will not generate the cores, what a traditional cores, 5%, 10% of the total what breeders know. We will develop a sort of pipeline. We call them iterative cores. For example, if we have 9,000 accessions of cysteine, you can screen all the 9,000 accessions in one go, you do it. If you, for some traits, can do only 100 of them, so you identify the most diverse 100 out of them, phenotype it. If you get a trait of your interest, use it. You don't need to bother with others. If you don't get it, you remove those 100 from this one, do the analysis, get another 100, and we call as iterative cores or the dynamic cores. And that's the philosophy how we have started using all these crops. And I'll give you some pictures of this one. See, this is mung bean, 4,100 accessions being grown at Kazari and at Badnampur. Yeah, master. Have a look on the page. All genetic resources, just one place. See the variability. This was for being background at two places at PAU Ludhiana and World Vegetable Center, Hyderabad. Simple, I'm just giving you one example of the variability in the earthworm. You have number of seeds per plot from as low as 3.6 to as high as 9. So that's a variability in case of the this one. Like how number of the seeds here, number of the primary plants are number of pods per plant. From somewhere as low, which is almost negligible, to almost 100. So this is the variability in the genome plasm. This is we have more data, 2 years data on this one. And all these genetic resources were grown at two places. And last year we invited the breeders from regional search around that, go and select whatever you are interested in, so that you can take that material directly. And one important thing has come for us was in cowpea. So cowpea, they had a problem of one of the diseases. They identified some stocks, and some of them are really kind of good. US Harvard has put 3 lines into the IBT this year, 4 of them, straight away. That's how you can really look how the genetic resources could be used correctly, science-based, so that you are looking for individual genes of this one. So under these projects, what we are seeing is that we will provide you the regions. For example, if you take this system, for example, if there are 20 lines which are resistant, we will, under this genomic-based project, will tell you whether these 9 have the same gene or they have the different genes. Gene. Right. Yeah. So that's being used. Oh, the cross one, right, sir. This one, 104. So this is the check. So this is the highest number so far checks. And you have the variability. So this is another paper. How this I'm telling you how the globally things are changing now. Now this is a wheat work now. The CIMMYT gene bank has 150,000 accessions of total, and they have analyzed 80,000 of those only last year. This paper appeared only a few weeks back. So this is now a complete analysis of the gene banks. Things are moving now. In fact, we were not knowing the gene banks, how much diversity is there, how much duplicity is there. All these high throughput genomics projects are going to give answers to many things. That how much is duplicity, how much that is being used. For example, here we see a lot of the land traces are still not in use into the regions. And this is 56,000 lines whose data is on. But this is for the breeders now. Now they know what are the lines which are the ones which have not been into the elite breeding lines and where they can be. And this is how the future gene banks are going to provide data to the breeders, to other researchers, so they can go more streamlined rather than hit and try method. So this was on the NBPGR, but if we have time, then I can give another 15 minutes to see how PAU did it using the genomics and wild species together. But otherwise, from NBPGR work, it's up to here. Okay, 10 minutes I can tell the people how this we did at PAU systematically to ensure that what we are getting. So we know that diversity bottlenecks happened at 2 places. One was the early domestication and second was the modernization. And then you have 2 routes, either get diversity back from these early domesticates to this one. And this is what we are doing at NBPGR now. But you have alternate way is that diversity we lost during domestication, you can bring them back. And this is what you can do from the wild species. And this is what we did at PAU. And I will give you the examples of rice sampling. Different species, and I will give you one example of rice that was bacterial blight. Bacterial blight was a major disease of rice. and not even a single known gene was resistant to all the virulences. Until that time, we screened 300 expressions of wild species, and we found a good number of them which were resistant to all the 7 pathotypes. The 7 pathotypes of the bacterial blight in Punjab at that time. Now there are 10, in fact. And we picked 1 expression from each of the species. We picked 1 from Nivara, 1 from Glabruma, 1 from Bartha, and 1 from Ruficola. And see, we are lucky enough in this one that the 3 we picked because our hypothesis was that they may be different genes. They may be same as well, but fortunately for us, the one we identified from Nivara is a different. We designated XA38. 1 from Glabruma we designated 46, and 1 from Ruficola this year submitted only last week. It came as XA47. So we are thinking in a very different way here. It's not only the basic research. We wanted to be sure that it's basic research, good publications, and material to the breeders' varieties straight away. And I will give you one example of XA38. From a wild species, we transferred this gene, mapped it, and that time rice was probably very good. But if we were dependent only on the linkage maps, FSSR-based linkage maps which are available, we would have been able to identify genes somewhere here. Not very good. But since rice genome was sequenced, a lot of information was available. We used the genome sequencing, other things, and ultimately we could identify a marker which is very closely linked to this gene, and which is a codominant marker which could be used for breeders. Now, this work took us 11 years to complete mapping. And how technology, if you stay with the technology, how the things will move. Until 2007, hoppers were not at all a problem in Punjab. 2007, this started becoming a problem down plant hopper. And we also started working on date 1. So what we did, this is a classical example of collaboration. Here you might have seen bacterial blight, a pathologist did all the screening. The breeders and molecular, they did the process, molecular biologists did their own work. So it's a collective work, not one person at work. Here our entomologists identified the ones which were really resistant to this hoppers, 2 years data, identified a few of them, and I will tell you one of the examples here of how we use this gene. So this was, we were going with the traditional method, and it might have taken us another 15 years. And again, thank you, sir, for providing us funds under the incentivization program. And month of March, when others were not ready to use, I came forward, I requested Dr. Sandhu that we are ready to use this model. He provided us good funding. By the same time, Dr. Singh had published this map. We used this map and we published this, we mapped this gene. So this is one PhD work, 3 years time, starting from first cross up to mapping, you had a gene. So that other one took us 11 years. And here is the vision of your people who are at the top. Dr. Sandhu had a vision. He knew this importance of this gene. He knew we didn't have the BPH resistance gene anywhere known, much of them. He really funded us well, and so this is so this is how the things actually move. And another gene, we started with the same, what we call now XA. Now this gene was, it took us 15 years actually to map this. It's not that we were not doing things with the systems. The classical SSR marker-based gene did not help us to really localize it. Until again, we had some, again from the same project, incentivization that continued. We had money, we got the GPS, and we could map this gene, and this is the thing, this is another XA46. This is the bacterial resistance gene. Likewise, we collaborated with, blast was not an issue for us until recently in Punjab. So we gave this interspecific material to TRR Hyderabad. Collaboration, we mapped this gene. Now, this is a one gene which is resistant both at the seedling stage as well as at the neck blast stage. And now neck blast has become a problem of basmati. So what was not envisioned 10 years back, that's a gene, so they are using now markers. This is how the collaborations will really help you. So likewise we have, and we have shared these genes, everyone published, Here this gene is CX38 being put by different colleagues, different groups into the varieties, and some of them are being released as the varieties now. So, yeah, I will stop here. So this is how the things are here, and all this work which I have presented is the collective work of NBPGR colleagues. I have presented their
So if there is any question, can audience can talk with us or interview, or within this group you can ask something about class.
So what is the strategy for conservation of populations?
So we have two ones. The cryoconservation is the important one, but all these have been conserved. In fact, cryoconservation we are thinking of not only using for the orthodox seeds. We would like to really use this technology for conserving all other seeds, especially the wild species. Because in seed banks, we have very stringent standards of having larger quantities of seeds, and wild species you don't get those number of seeds. Another thing with the seed banks, really have to rejuvenate them. Some maybe after 30 years, some 40 years, but you have to rejuvenate. And in cryoconservation, probably you may not need to do it after carbon age. So cryoconservation, we plan to really look for different way other than the unorthodox systems.
So another thing that you are doing the conservation through pollen culture. But as per my opinion, the low cytoplasmic concentration may create the problem with conservation. However, the LSCs have reaching the cytoplasm. What kind of difficulty we are facing in the conservation?
For pollen cryoconservation, you will have two advantages. One, I am propagating this technology for the horticulture treatments, where you have the tree species if you really want to undertake any crossing. Many times there's no niching of the flowering of the two different accessions, the two different varieties. So you can conserve them in liquid nitrogen for a short term, maybe a week, 2 weeks, 3 weeks. So. And second is especially the wild species, which we may lose for some time. If we can really collect a large quantities of the pollen, and here, of course, one has to be not a small, a few hundred vials of each of them, conserve them. Maybe 20 years down the line, someone may like to have, oh, that was a species growing somewhere, I would have loved to cross them. So you have two options. One, you can regenerate from pollen plant, applied plant back. They can be difficult, but you just using for pollinations, one should be able to use this one. and we have found that very successful right now. Especially we found it in wild species of okra, which crossing with the niching of the flowering is very difficult. And if you have the pollen conserved, the liquid nitrogen, you can use that. So we have good results coming out of that.
मेरी क्वेश्चन बहुत लेमन की तरह है, फार्मर्स और हमारे स्टूडेंट और यहाँ मीडिया पर्सन भी बैठे हुए हैं। अगर हम घूमते फिरते कोई भी स्ट्रेंज प्लांट या ऑफ टाइप प्लांट या बिल्कुल डिफरेंट प्लांट नजर आता है जो कि और पॉपुलेशन से भिन्न है, हम उसे आप तक कैसे पहुंचाएं? उसमें भी कोई यूटिलिटी हो सकती है, कोई भी रेजिस्टेंस हो सकता है, कोई वाइड स्पीसीज हो सकता है।
हर एक को, जितने भी आपके स्टूडेंट्स हैं जो सुन रहे हैं, चाहे अंडर ग्रेजुएट है, पोस्ट ग्रेजुएट, जो गांव से हैं, उनको लगता है कोई भी चीज अलग से है, उसका सीड जरूर कलेक्ट कीजिए, हमें डायरेक्ट भेज दीजिए। हम आपसे दो चीजें चाहेंगे उससे कि अगर आपके पास जीपीएस डेटा है, जो आजकल मोबाइल्स में है, ठीक है, नहीं है तो गांव का नाम जरूर लिखिए, गांव और जिले का नाम जरूर लिखिए। उसके बाद में गांव का जिले का नाम है, सारी इंफॉर्मेशन वहां से दे सकते हैं। तो वहां से अगर हमें लगता है सीट कम है या कुछ है, तो हम दोबारा भी वहां जा सकते हैं उनके बिना भी उसको कलेक्शन करने की। बट हमारी जनरली ये है कि जीन बैंक हम चाहते हैं कि हमें जो लॉन्ग टर्म स्टोरेज के लिए है, सेल्फ पॉलिनेटेड क्रॉप्स के लिए हम 2000 दाना चाहते हैं कम से कम। अगर है तो ठीक है, नहीं तो हम उसको इंक्रीज कर लेंगे। ये किसी पर भी कोई लिमिटेशन नहीं है कि हमें 10 दाने भेजे तो हम नहीं लेंगे, नहीं, हम लेंगे, पर उसको नंबरिंग, जो नेशनल नंबरिंग है, वो मिलेगी तभी जब उसको लॉन्ग टर्म सीट पे जाएगा, वो तब जाएगा जब उसको सीट इंक्रीज कर सके। यस, हमें क्रॉप का नाम बता दीजिए, कहाँ से कलेक्ट किया है, किसने कलेक्ट किया है, और कलेक्टर का नाम हमेशा रिकॉर्ड में रहेगा, ये जरूरी है।
I am Dr. N.V. Gupta, plant physiologist. So first of all, I extend my thanks to you for coming here and giving a good lecture. Sir, I have two questions. Not question, I need advice. Sir, I got a project in 2010 on wheat for developing high temperature tolerance method for identification of temperature tolerance method in wheat. And for that, we collected 102 accesses from NBPGR, and we agreed an MTA we have signed. That research at that time we have the industry in Bikaner. So sir, from 102 accesses we narrowed down every year, and in first year we narrowed down to 45 and 11, and now we have 3 accesses which are highly tolerant to heat tolerance. So how we can use this material? And my second question is that sir, one of my colleagues in Australia, he has started working on the spelta wheat, Triticum spelta, and it is highly tolerant to drought. And last week, I am in communication with him, and they have developed a rinse of the spelta is also hydroploid, and they have developed 8 to 10 rinse, and they have identified the drought tolerant genes, and TA, DR1 and DR2 gene has been identified through an SMPs they have developed, and actually we have developed a paper also. So if we want to collaborate with them, how NBPGR can help?
Coming to your first question, that very happy to see that you identified 3 lines. See, if you see the MTA, NBPGR doesn't ask anything except that if anyone is commercializing it for financial gains, then those financial gains will be shared between ICAR and the university. As of today, we are not working on those lines. It's a basically seva hi hai aajkal jitne bhi universities hain, it goes free funded to the farmers. So I don't see any difficulty as long as you are acknowledging the use of genetic stocks. You can use that for publications. Acknowledge, keep the original identity of them. That's most important for us to see. MTA में कोई strong चीज नहीं मानता। हाँ, अगर आप commercialize करेंगे, अगर आप पैसा कमाएंगे, तो फिर ICAR भी उसका हिस्सेदार होगा, वो ऐसा होता है, ठीक है? This is simple, and of course हम पैसा कमाने के चक्कर में भी नहीं है, क्योंकि farmers को भी हम सब कुछ फ्री दे रहे हैं। Coming to your next question for spelter. This delta was from India or from Australia. So you have to sign MTA with them. As you can bring that seed through SMTA.
No, sir. I have got there, and now at the time we were on a birthday. Now they are shifting to Spelta, and they want to work with us.
Yes, so you can have a collaborative projects. That's one thing. Otherwise, getting germplasm from them to this place is absolutely no problem. You can simply send an SMTA to them. You send an import permit and get the seed from them.
So directly or through NBPGR?
No, through NBPGR. Through NBPGR.
You have to get the this import permit from them. Then only you have to.
So you simply send a request to NBPGR. What germplasm you want to get it. We will introduce. We will send you an import permit. With them, you have to sign an SMT because this is what I told you. The treaty. So all the global exchange is under International Treaty on Plant and Resources for Food and Agriculture, and this is NTA, what's called a Standard Material Transparency Code NTA. So you sign that one, because they will require that one. Who will issue you the import permit, you get this.
मेरा पहला क्वेश्चन ये था कि जब हमने आइडेंटिफाई द वैल्यू आप उनका विजुअलाइजेशन सर कंप्यूटिंग का करते हैं ये कहीं पर प्लेटफॉर्म का Sir, over the years of evaluation, we have identified 3 lines out of 200 by 102. Identified, so you have the sufficient data. Yes, sir. How many years data? Years data. So first of all, you can do one thing. You can get the list of these 3 lines from NBPGR. So these lines will come to the under public domain. Yes, then the readers can utilize. So first of all, your or my duty will be to register these NBPGR with NBPGR. So then the value will be available.
I can suggest you that the fastest go to NBPGR web page. It's an online submission, right? What are the requirements? Submit that one, and our next meeting for registration will be first week of December. So you can get the fastest registration of this stuff.
Do you need an endorsement of Institute Employment Registration Committee?
Yes.
We have to pass that on at the institution.
You registered that as a particular stuff.
But you belong to this university.
Good morning, sir. Sir, this is Dr. Varsha Kumari. I have one question. Sir, I am in search of wild species of fenugreek resistant to powdery mildew. So how can I get from NBPGR?
You are looking for wheat which is resistant to powdery mildew?
Fenugreek. In fenugreek, the wild species I'm searching for that is having resistant to powdery mildew.
Okay, so okay. We have put actually the proposals are under in the preparatory phase. All these seed spices that Kuldeep will be a partner for Methi in that one. ऐसा मुझे नहीं पता भी हमारे पास कोई रेजिस्टेंट सोर्स है इसका भी कि नहीं है. I'm not aware today that if we have a line which is resistant to powdery mildew or not, we will look it, make a note it, and if there are some lines which are known resistant, then we will inform you. Else, what we can do is, whatever germplasm we have, you can take it and screen it at the hotspot first and identify the one which is resistant.
Yes, sir. Just I wanted some wild species. I can go for screening of it. Just I wanted to have some wild species of fenugreek.
Okay. We have made a note of this one, and we will inform you what wild species we have. We will inform you through Dean, Director of Research, will inform to you. We'll inform you through research, maybe by tomorrow evening or day after tomorrow, we'll inform you about it. And whatever germplasm is available, you can take that one, and there's a standard procedure for that one, and use it.
Thank you so much.
Thank you very much, sir. Sir, it's nostalgia. Let me submit that today's lecture is in the series of lectures we have been organizing since the inception of National Agriculture Higher Education Project at Sri Agriculture University, Job. To name few of the dignitaries, we had opportunity to listen to a technocrat turned bureaucrat, Mr. Ashok Dalwai, then Padma Shri Honorable Dr. R.S. Paroda, Dr. Siddiq from Australia, Dr. Bariana, Dr. Kulwinder Singh, and today we have with us our own Dr. Kuldeep Singh from NBPGR. So thank you very much, sir, for accepting our request and giving a very scintillating lecture that benefited our scientists, our students, and farmers who were connected with this program. I am equally thankful to Dr. Ashok Kumar, Principal Scientist and Head Department of Germ Plant. Rashmi ma'am for sparing her valuable time and giving us a lot of insight into the subject at the CSIMM trial site. I am highly thankful to Honorable Vice Chancellor sir for guiding us in organizing such lectures and inviting dignitaries from different institutions and organizations. Thank you very much, sir.
Now I request our Honorable Vice Chancellor for presentation of Gamcha in the memory of Dr. Kuldeep Singh, arrival at Alma Mater.
I worked in ICAR, I have to inherit the name.
I request Dr. Yake Bhutan to present a Gamcha to Dr. Ashok Kumar. Now I request Dr. Rathod to present a Gamcha to Dr. Rashmi Yadav. Thank you, sir. Once again, thanks.